This video is lots of phospho and AKT. So warning, there's some really long names here, and the most important thing is that you know the abbreviations of these. You don't need to know their full names. You also do not need to know their structures. So the most important thing is to keep track of what is the reactant or substrate, what's the enzyme, and what's the product. So we're going to start off with P13K. And so this is a enzyme, which the full name is here, phosphoinositide 3 kinase, also known as phosphatidylinositol 3 kinase. And basically, you will see this is the name in the textbook. Most of the time, I'm going to use these names, the phosphatidylinositol. And so the phosphatidylinositol 3 kinase, P13K, that's this green blob right here. So let's draw a big green blob around this. And basically, as an enzyme, it's going to take a reactant, which is PIP2, and it's going to convert it into a product, which is PIP3. PIP2 is a phospholipid. This is located in the plasma membrane, or PM. And notice that PIP2, it's really highlighting the 2, and so that has to do with 2 phosphates that are located within the sugar ring. PIP2 is also known as phosphatidylinositol 4,5-bisphosphate. If you're not familiar with this sort of naming, bis means 2. So there's 2 phosphate groups here. And I just mentioned this because it might help you keep track of what's happening here. So we start with 2 phosphates on this phospholipid. This enzyme P13K adds another phosphate, and now we end up with PIP3. So now there's 1, 2, 3 phosphates, which are amongst the core of this sugar molecule. Let's talk a little bit about PIP3. So PIP3's full name is written here, phosphatidylinositol 3,4,5-trisphosphate. So the tri here, referring to the 3 different phosphates here. And basically you can see there's more complicated structure here. This is the sugar group. There's a glycerol kind of backbone here, and then we've got fatty acid chains here. That's the case over here for PIP2 as well. Those are some clues that this is a phospholipid. And basically PIP3 can recruit a protein kinase, which is this part here. Specifically, it's called protein kinase B, PKB. There's a whole lot of protein kinases floating around, so they actually usually refer to this as ACT, AKT, and so that's how I'll refer to it. But I've written out this information about its name so that you know the following, that ACT is a protein kinase and it signals for survival and growth. On the next slide, you're going to see more about this ACT protein kinase, and it's a green blob. It kind of looks like this. It's a green blob, and I'm just going to draw it kind of like this, although it's got a little bit more kind of a squirrely kind of thing here on the top. Fun fact for you, this name AKT, that actually comes from a viral gene. So AKT is pretty similar in terms of its sequence to a viral gene. And basically this ACT protein kinase is what PIP3 will activate, and so that's what's going on over here, is that PIP3 will activate, if you will, growth and survival through this ACT protein. So this figure is from your textbook, and it shows you a little more about the details, in particular the mechanism of how RTKs are going to activate P13K and AKT. And so they kind of put a little dash here because these will work together as a complex to relay the signal. This is figure 16-35 in your textbook. So let's kind of break this down. Basically, over here we see this activated receptor tyrosine kinase, and so there's a ligand that has bound, which they're calling a survival signal. And basically, as you know, receptor tyrosine kinases will trigger phosphorylation events and activate proteins. And over here what we see is an activated P13K or a P13 kinase. On the last slide, this was shown in green. And this was the kinase, which was taking the PIP2 as a reactant and generating PIP3, which is over here. So let's label those. This guy is PIP2 and this one is PIP3. Again, PIP2 being this membrane-bound molecule with the two phosphates, and then PIP3, the product of P13K's activity, where it now has 1, 2, 3 phosphates, and we call it PIP3. It's also membrane-bound, as you can see through these covalently linked lipids. And then in your textbook, there's actually another step over here, another step over here, which I'm just going to ignore. There's lots of details about how these things happen, and what I want you guys to see is the overall theme, which is that our receptor tyrosine kinase activates another kinase and then will convert a phospholipid into another phosphorylated form, PEP3, and then that will activate AKT. So that's what's going on over here. So AKT is this green blob, kind of looks like it has antlers, and it is in its activated form, as we could surmise from its little pink lines here. And you can see that it's got some phosphate groups here, so it has been phosphorylated. I notice that it is evidently interacting with PIP3. So basically ACT has been recruited to the membrane by PIP3. Due to this physical interaction, sometimes we call it a docking site. So ACT, which is normally just kind of floating around in the cytoplasm, now docks here at the membrane through PIP3. And then basically this interaction between these two will lead on to the next step, and that signals the downstream effectors and allows the relay of signals to take place. And so I've broken down the mechanism down here. So basically, going through step by step, there's a survival signal, which activates the RTK that recruits P13K right here, which is then activated. So this becomes its active form. That means then it can phosphorylate PIP2 and make it into PIP3, which is this part right here. Then PIP3 is going to be an active docking site for AKT, which is then recruited to the membrane through this phosphorylation, and then it stimulates downstream effectors. One more thing I want to mention is that these survival signals can often be growth factors, and in particular one of them might be insulin growth factor. So now that we've seen how AKT can be activated, I want to talk about two different roles that it has. AKT can repress a protein called BAD or BAD, and this protein would normally lead to apoptosis, which is a type of programmed cell death. And basically, what that means is that when AKT represses BAD, it is also repressing apoptosis. At the same time, AKT will activate a molecule called TOR. And TOR has two different roles. TOR stimulates protein synthesis, in other words, translation, and it represses protein degradation, which would be the breakdown of proteins. Both of these processes are required for cell growth. And so basically, when AKT activates TOR, it is also activating cell growth. So it's kind of cool as you can have one component of the signaling pathway that can essentially distribute roles to suppress cells dying and also encourage cells to grow. Another kind of fun fact about this is that rapamycin, which is an anti-cancer drug, also can clue into this pathway. So rapamycin actually represses TOR. And so essentially what it does, because an anti-cancer drug would be suppressing cell growth, it prevents TOR, which would normally lead to cell growth. So I don't expect you guys to memorize this pathway, but I do expect that if I provide you with a pathway of the names of these molecules here, that you'll be able to assign the roles. So know that BAD leads to apoptosis and TOR leads to cell growth, and basically be able to interpret that pathway. Using kind of this core information, which is here, add on the extra parts, this and this, and be able to predict what would happen if we messed around with this by having mutant or constitutively active versions of these proteins.